The carbon dioxide partial pressures, that is how it affects. If they ask you explanations, students should be comprehensive to that details. So we shall now be sure of the quality of students we are entrusting with our environment. Another factor is the temperature. You know these factors happen for photosynthesis, you know them very well again. The elementary biology of temperature, students will always mention, during high temperatures, the rate of photosynthesis is high. During low temperatures, the rate of photosynthesis is low. Can you explain why? Do you take time to explain why temperatures have that variation of control, of productivity, or of control of photosynthesis? I want to emphasize from the juvenile biology that we studied earlier, there is a factor they taught you about enzymes. Enzymes and their behaviors. The behavior of enzymes are known that for them they are sensitive to changes in temperatures. That should be in your head. Because enzymes control all the biological processes in the living organism, it is true that temperature must be fundamental in these living organisms. During the COVID-19 period, physiological scientists advise that you should take temperature, body temperature of all living organisms to ensure they don't deviate from normal. Which it means that there is a negative effect if deviation occurs from normal, whether a negative deviation or positive deviation. That implicates that maintenance of the norm is fundamental. We must ensure the temperature remains at that level that allows the body cells perform to their best. Likewise, living plants are living organisms. Some of you undermine them, but trust me, plants have enzymes like you have in your body. I'll tell you an interesting one, an interesting scientific aspect that will open your eyes more wider toward the scientific view. I happen to have a sister prepared the juice for the father, the lemon juice. The sister was supposed to crush lemon, probably oranges and carrots and mangoes. He extracted the tonic juice. This was too fresh for consumption by the father, juice A. The same sister prepared set B, but this time around, he crushed the same fruits, fresh fruits. He used the hot water to prepare this juice. Use the hot combination of water there and he poured in this juice. Then the father consumed the glass. The father consumed the glass on juice A. And he also consumed the glass on juice B. He left some on each set. Now the question comes, teasing question. The juice remained for A and the one remains for B. Which one will go bad first? And why? Another one is food for thought. Technically, you realize that glycine A will go bad in the next few hours. In fact, if you are involved in pineapples here, it will go bad even much more faster. And the glycine set B, you can even retain it for 2 days when all factors in the two sides are kept constant. Even if you are stored the A and B in the fridge, A will go bad faster, very fast, and in B you can retain its life for more days. The explanation for this is relying on enzymes present in the cells of these fruits that you crushed. I want to mention that these fruits, they are not fruits just that you extract them and you crush them. This mango, it is made up of several cells, and these cells here, we call them amylopectin. They are known, they are a type of parenchyma cells, cells responsible for storage. They are all over the plants where storage of food occur, in the cassava, in the sweet potato, Irish potato, sugar cane, fruits, they dominate there. The ovary is made up of a few myelopectin cells of the parenchyma, but the more flowering process occur and you have successful fertilization in this flower taking place, these cells will start mitotically dividing and they become many. Because they are sure, they are going to store a lot of food that is required for propagation of this seed. So whenever you see a mango, you wish to pick it. That's why they had to store this food, because they thought wisely, these plants, they thought wisely that you will need this food. And when eating the food, for them, you will help them propagate their life. So you carry this seed. Probably you wish to carry these mangoes from one locality to another, because you might want to finish them from the plant where you picked them. And that allows their evolutionary success to colonize new areas. They are increasing advantages of colonizing new areas, so in case you throw them away on the dustbin, they will acquire atmosphere of germination and they will grow, they will acquire success. Enzymes present in these cells where starch is stored and these sugars that are ripening, those enzymes present there, they are the ones that are going to kill your juice if you prepare it with the lukewarm or probably water of low temperatures or atmospheric temperatures. These enzymes are going to be provided with a suitable temperature that allows hydrolysis. Here of these chemical compounds. Science is too broad. If you covered your cytology, I want to remind you of a property that this cell has. This cell has got a nucleus, which is porous, nucleoplasm, nucleolus, present with mitochondria, all of those are organelles. Remember the Golgi body apparatus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the ribosomes, the lysosomes. These are very important. And you are aware that enzyme are produced by protein synthesis. Because all enzymes are proteins in nature, you are aware. These lysosomes have a behavior. These lysosomes are saying that they are enzyme bag. For them, they are waiting. Any cell organelle that is misbehaving, these lysosomes go and fuse to that cell organelle misbehaving and they destroy it. That one is called autophagy. They'll ask you, what is meant by autophagy. The process by which hydrolytic enzymes break down worn out cell organelles. But now, these lysosomes will form autolysis. If we happen to have a cell itself misbehaving, there are going to come a situation when a cell is misbehaving. Lysosomes are going to break down that cell. The lysosomes are going to burst within the cell cytosol. Cytosol is the fluid cavity that is inside this cell here. When they distribute themselves all over, it means that they are going to carry out hydrolysis of all. They are going to break it down, to catalyze the breakdown of all the organic compounds in this cell here, and eventually the entire cell will be broken. That's why you say autolysis. Lysis to mean breakdown. Auto cells. The cell will self-digest and it will disappear. That is on cells which are misbehaving. Cells of the red blood cells sometimes remember they misbehave. When a cell develops some radioactive substances, those cells happen to misbehave, and when they misbehave, the body the body realizes they are risky cells to become carcinogenic cells or to become cancerous cells. So before they multiply, the lysosomes in those cells will burst immediately to have these cells digested and destroyed. That's what they say to be autolysis. Now, if you were branding these cells of the mango to form a juice, the lysosomes will be so sensitive and wise to realize that now these cells are destroyed, and probably they are risking to cause harm to the plant. Because plant cells have elongated, they have relatively wide range of period of life, they will still be responding even when they are removed from the plant. Lysosomes will therefore be active in this juice, trying to break down not only the juice but even the other chemical compounds present to have them change into a new chemical form that will not be tested very well to you. You will describe it as this has gone bad. So technology is able to solve this problem here. Scientists that have understood the food science and technology, they have realized it is wise when you're making grease, use the hot water. Because these enzymes, or whatsoever lysosomes present in those cells, they are sensitive to changes in temperatures. And when the temperatures are very high to them, they are denatured. Their active chemical structure is altered. And when you alter their active chemical structure, you will appreciate that they cannot work anymore. You will therefore keep your juice for that long, not until you become susceptible to bacteria and they come to introduce new enzymes there. But if you are kept warming it even after 2 days, you can even keep it for a week. Because that will keep not favoring the newly introduced bacteria. If you assume that this is temperature, this graph here, and probably this is the rate of reaction, consider enzymatically controlled reaction. You realize that at low temperatures, maybe 0 degrees Celsius, the rate of enzymatic reaction was seen low, and then it increased rapidly, increase in temperature. It was at maximum reaction at the peak when temperatures was at its best optimum. That is 40 degrees Celsius, best optimum. We are always worried whenever the temperature gets off the normal, at 7 degrees Celsius in the living cells, and it goes beyond 40, you're going to die. Cells are going to boil. If the temperature goes beyond 40, definitely the rate of reaction is going to decrease rapidly, and reasons are very clear. I say that the enzymes were denatured, their three-dimensional structure is altered, they will be unable to work because their active site is destroyed. If that one can explain why these enzymes in the mango juice were able to be destroyed and create a lot of life for this juice without getting bad. The plants have these enzymes and they are controlled by temperatures in action. If you remember, when I introduced an aspect of C4 and C3 plants, I was very clear. I mentioned the differences that C4 plants remember that their optimum temperature of operation is relatively, if you compare it with the other one, it is relatively high. That for it up to 40 degrees Celsius, cells are still functioning. The C3 plants, by just 38 degrees Celsius, they are failing to sustain this temperature. So enzymes here, enzymes beyond this temperature, enzymes denatured. Destroyed. So you want to tell me that the rate of productivity in C4 plants will equal to that in C3 plants given this factor of temperature? No. C4 plants will play with high productivity and C3 plants will show low productivity, and this is because the C4 enzymes in C4 plants, they have a high optimum temperature. They have a higher optimum temperature compared to that one in C3 plants. However, both of them, the rate of productivity will be low. In both of them, the rate of productivity will be low when temperatures are low. Why? Enzymes are inactive at a low temperature. Enzymes are inactive at a very low temperature. They cannot function. They will freeze. That's how temperature affects the rate of productivity. If I ask you to explain. Number three is consider water. Is water one of the factors affecting the productivity? You remember how water is important? Explain the details on how photolysis occur, and then talk about the hydrogen ions produced and the hydroxyl ions. So hydrogen ions is known to lower the pH. So talk about in that perspective of amount of water provide hydrogen protones that reduce the NADP, which provides the reactants for the dark stage, because the NADPH2 normally utilizes it. Remember, we use the NADPH2 to reduce the phosphoglyceric acid. This is number 3. Number 4, sunlight intensity. And then I'll stop on this one. You'll read about the details, the other factors and their explanations relevant to them in your notes. Light intensity. I want to mention that light intensity, how does it affect? High productivity, we expect high light. Low productivity, we expect low light intensity. Students who understand the compensation point will appreciate that. When light intensity is low, we expect the rate of productivity to be low, and when light intensity is high, expect the rate of productivity to be high. The more we increase light intensity, we are increasing productivity. So what is happening here is that sunlight intensity, the reasoning is that it is known to strike the photosystems. Remember that one during the life stage? Cause them get excited after gaining kinetic energy. So the kinetic energy they get will depend on how much sunlight has struck them. So that will determine the number of electrons we excite high energy level to be accepted by electron acceptors, and that will determine now, you see, it will determine how far these electrons can go. And the farther they go will translate the amount of energy they release when they are coming back to low level. Automatically we shall use that energy to bind an ATP and an organic phosphate to produce an ATP molecule like that. That's said during starlight energy. This is actually said to be photophosphorylation. We're appreciating that previously. You see, if we have too much energy, sunlight intensity is very high, we shall have several sets of ATP and inorganic phosphate binding together to produce ATP. Then when we have little energy, so the sunlight intensity will determine how much energy is present when electrons coming from high energy levels. So whenever electrons are coming to a low energy level, expect little energy released. When they are coming from high energy level, a lot of energy will be released. So number of ATP molecules will be formed in that way. This is supposed to mean high light intensity determine the kinetic energy and how far the electrons that are excited can go. Now that is the explanation. That will explain how light intensity affects productivity. Because we simply need light to generate ATP and generate NADP H2. You remember that from our previous episode when we were understanding photophosphorylation. Products of light stage are ATP and NADP, and we need them in the darker stage. That is all. So we need light to excite electrons at the photosystems, and we need light to ensure water molecule undergo photolysis, producing NADP H2. So the higher the light intensity, we are going to produce much of these products. And when they are very many, we are most likely to have a variety of phosphoglyceric acid. We are most likely to have a variety of phosphoglyceric acid converted to a stable sugar. This is unstable. A stable sugar, phosphoglyceraldehyde. I say that here we need NADP H2, and then we produce NADP to discharge, which means that hydrogen ions are left inside. And in the same vein, we have ATP molecule formed from ATP and inorganic phosphate. So the more we have these products, we are going to have the equation shifting more to the right, shall produce these products, a lot of them. And this one here is what we call increased productivity. We definitely, if we have a lot of these, we are going to expect sugar molecules formed, many of them, glucose molecules. Because whenever these accumulates, accumulation of these molecules, and they become very many, maybe like 13 or 20, or probably whatever number you can choose to say, we are going to have an equivalent exhaust sugars generated during conjugation reaction, and then we'll be this Rubik, we need it for sustainability of the reaction, because this reaction cannot proceed if Rubik was not present. That I want to rest my case on that. And then I'll have to introduce